There is a stigma around STIs. Even with perfect hygiene, people are still getting UTIs. Especially Black women have been subjected. They've been used, they've been studied, they've been cut open and experimented on without consent. Period pain is normal, but period pain that interferes with your daily life is not. What if the reason you haven't gotten answers isn't because your body is broken, but because the system was never built to listen to you? Welcome to G-Chats. I'm your biomedical engineering big sister, and welcome back for another episode of G-Chats with your host, Gianna Jara, sitting here in the G-Spot. I'm super excited for today's episode because it is something that you probably see all over the internet. It has become a huge topic online and in women's health spaces, and that is none other than ureaplasma. And there's so much information that is misinformation. Sometimes people don't understand it fully, and you might have heard people saying things like ureaplasma is an STI or that ureaplasma is a hidden infection causing all of your symptoms. There are a lot of truths to some of the things said online, and truthfully, the reality is that the conversation around ureaplasma is much more nuanced than people want to make it out to be. And so in this episode, we're going to slow down and unpack what ureaplasma actually is and why it behaves differently from many other bacteria and why it can sometimes be involved in symptoms, even though it is often a completely normal part of the human microbiome. So let's talk about it. So first, let's discuss what ureaplasma actually is. And if you are watching on like YouTube, you will see me looking down. I basically gathered all of the scientific points that I want to talk about. So I'm looking down at my iPad so that I can follow the talking points correctly. Ureaplasma is a genus of bacteria that belongs to the group Mycoplasma taceae. And so, or Taceae, Taceae, I've heard it said many different ways, but essentially this is the group, the family of bacteria. And it includes both Mycoplasma species that you probably are all aware of and Ureaplasma species. And the two Ureaplasma species that are most commonly found in humans are Ureaplasma parvum and Ureaplasma urealyticum. So these are the two most common ones that are talked about when we're talking about women's health. And what makes these organisms particularly unique in a microbiological standpoint is that they do not have a cell wall. And that is super important to understand because most bacteria have a rigid cell wall structure that helps maintain their shape and it protects them from environmental stress. Well, ureaplasma does not have that. Instead, it only has a plasma membrane, and this structural difference is super important for two reasons. Number one, it means that ureaplasma is naturally resistant to certain antibiotics that specifically target bacterial cell walls, such as beta-lactams, and that includes penicillin, amoxicillin. So it's naturally resistant to those antibiotics. And second, the lack of a cell wall makes these organisms extremely small and flexible, allowing them to live closely associated with host cells in mucosal environments. And why that matters is because the vagina is a mucosal environment. And so another defining feature of ureaplasma is in the name itself. So the urea in ureaplasma refers to the organism's ability to break down urea, which is a nitrogen-containing compound found in urine and in bodily fluids. And ureaplasma produces an enzyme called urease, which allows it to metabolize urea as an energy source. So this metabolic trait is one of the main reasons ureaplasma is commonly found in the genitourinary tract, where urea is present. Now, this is where things get super interesting because ureaplasma is often described as a commensal organism, and the word commensal is a microbiology term that we use to describe microorganisms that live in or on the human body without causing any harm. And in fact, many commensal microbes can be beneficial or neutral. We have plenty of bacteria that's commensal, and they are a forming part of what we call in microbiology the microbiome, because the microbiome has so many different types of bacteria, and commensal is one of the big types of bacteria in the microbiome. And our bodies are full of bacteria that are simply living alongside us and don't do anything. So these aren't foreign invaders and foreign pathogens in the traditional sense, and their mere presence, like presence alone of ureaplasma in your vagina or in your urine, does not mean disease. But this is exactly why ureaplasma is so confusing, because while it is commensal, studies have found that ureaplasma can be present in a significant percentage of healthy asymptomatic adults. So many people carry ureaplasma without ever experiencing any type of symptoms. And because of this, most clinical guidelines do not recommend routine screening for ureaplasma. Why would they? Because ureaplasma is seen as a normal commensal bacteria. They're like, every woman is going to show positive for ureaplasma. So why even test for it? Why even look for it? That's the logic in the minds of a clinical physician or a physician in general, just somebody working in that space. So from a clinical perspective, detecting ureaplasma does not automatically mean that there's a problem. This is important because the story usually gets super messy right here, because just because a microbe is commensal and it can exist harmlessly does not mean that it will always be harmless. And this is where I say some bacteria is just straight commensal, and then other bacteria is commensal but also opportunistic, because the human microbiome is super dynamic, and when that ecosystem becomes imbalanced, organisms that are normally quiet residents, commensal, can sometimes begin to thrive in ways that contribute to symptoms. So this is why I call them, and many microbiologists will refer to them as opportunistic pathogens, because when something is opportunistic, it usually doesn't do anything unless it's given the opportunity. And that opportunity is usually when there's an imbalance within the microbiome. So to understand that, we have to zoom out and we need to look at the vaginal microbiome more broadly. So in a healthy state, the vaginal microbiome is typically dominated by lactobacillus species, and these lactobacillus species produce lactic acid and help maintain an acidic environment. This acidity helps suppress the growth of many other organisms, not just ureaplasma, but many different organisms. And when that balance shifts for a number of different reasons, it could be for the use of antibiotics because you had a sinus infection, it could be stress, your hormones, you're going through menopause, it could be your postpartum, it could be literally many things. And we can always make a podcast episode all about the things that cause people to have microbiome imbalances and pH imbalances inside of the vagina. But there's many different things that can shift that balance. And when that balance shifts, we can see conditions like bacterial vaginosis, which is abbreviated as BV, which most of you probably know about. And this is where anaerobic bacteria begins to outcompete protective lactobacilli. And another imbalance that we also see, and that's sometimes discussed, is aerobic vaginitis. And this is abbreviated to AV, and it involves a different group of bacteria. So it's aerobic organisms like E. coli, Enterococcus, and staph. And that's why this is called aerobic vaginitis, because it's aerobic bacteria, and then bacterial vaginosis is anaerobic pathogens. Altogether, these imbalances can produce symptoms such as unusual discharge, irritation, burning, or odor. And when someone presents these symptoms in a doctor's office, healthcare providers are typically going to test for the most well-recognized causes, and that usually includes BV, yeast infections, aerobic vaginitis, and or sexually transmitted infections like chlamydia, gonorrhea, and trich. That's the key. And you probably noticed that I didn't include ureaplasma, because providers are not going to include ureaplasma in a routine testing panel because it is not seen as a true infection for many providers, because how commonly found it is in pretty much all genitourinary tracts. So one reason for that, again, is because of the commensal nature that we talked about, because ureaplasma can be present in healthy individuals without causing any disease. So testing for it can be difficult to interpret, because if a test comes back positive, it doesn't necessarily mean that it is responsible for the symptoms. So clinicians a lot of times have to weigh the difference, or whether it is simply present or if it's actually contributing to disease or pathology. Another reason why a lot of physicians will opt to not test for ureaplasma is because it can be challenging to detect because it's about how it grows in the laboratory. So traditional bacteria, when I worked in a microbiology lab, traditional bacterial detection often relies on culturing organisms, meaning that the sample is placed in a growth medium and then we allow it to multiply so that it can be identified by laboratory scientists and laboratory technologists. But ureaplasma grows very slowly and it requires a very specialized medium, and that's because of its unique biology and its lack of a cell wall, and its small, very tiny genome makes it more fragile and harder to culture than pretty much all other bacteria. So as a result, standard cultures end up missing it. So then this is where modern testing methods come into place, and we're specifically talking about PCR testing. So that stands for polymerase chain reaction, and PCR is a molecular technique that detects the genetic material of microorganisms rather than relying on their ability to grow in a culture. So instead of waiting for bacteria to multiply in some lab dish and in an incubator, PCR amplifies small fragments of DNA so that they can be detected even if only a tiny amount is present in the sample. And because of this, PCR testing is significantly more sensitive for organisms like ureaplasma. So many of the tests that do identify ureaplasma today are nucleic acid amplification tests, can detect both of the major types of ureaplasma, Ureaplasma parvum and Ureaplasma urealyticum. This increased sensitivity also introduces another challenge. PCR can detect very small amounts of microbial DNA, raises the question that is the organism actually causing disease, or is it simply a part of the background noise? And that's why it's very difficult, because no physician is going to culture it. They're just going to run a PCR test. But if it shows up positive, is it actually causing the problem? Because it is a normal part of the healthy microbiome in many cases. So this is why ureaplasma is often seen as a co-infection, and it's more important to test for this when the microbiome is imbalanced. Now, if you're a healthy, balanced vaginal microbiome, looking for ureaplasma is kind of useless in a healthy vaginal microbiome. But when symptoms are present, it can be a co-infection. And that is important to understand. And that's what happens when someone does have symptoms and is treated for something super common like bacterial vaginosis. The issue is that ureaplasma isn't tested for, and BV is most often treated with antibiotics such as metronidazole or clindamycin, which target anaerobic associated bacteria within the BV community. And these medications are effective against organisms like Gardnerella and BV-associated microbes. Those antibiotics do not particularly work against pathogens like ureaplasma or bacteria, microbes like ureaplasma, because ureaplasma lacks a cell wall. And because of its unique metabolic characteristics, the antibiotics typically used for BV do not reliably eradicate ureaplasma. So ureaplasma is generally more susceptible to antibiotics that target protein synthesis, such as macrolides, and this is used rather than the drugs typically used for BV. Imagine a situation where someone has symptoms, they're tested for very common causes like BV, and then turns out they're positive for BV. The provider is going to assume the reason they're having an infection is because of bacterial vaginosis, so then they're treated with metronidazole, right? That's the first line of treatment. If ureaplasma is also present and contributing to symptoms, that BV treatment might improve some aspects of the imbalance, right? Reducing the amount of anaerobic bacteria, but it leaves the ureaplasma population largely unaffected. So then this can create a frustrating loop for many patients because symptoms may persist even after treatment for the most obvious diagnosis. So at the same time, it's also important not to swing too far in the other direction and assume ureaplasma is always the hidden culprit. Again, many people carry it without obvious symptoms or any symptoms at all. So interpretation has to be very careful and individualized, and you need a physician who's up to date and modernized with this information, who's at least willing to rule out if ureaplasma is a factor. So ultimately, ureaplasma sits in a complicated space in medicine. It is neither a classic harmless microbe nor a universally recognized pathogen. Instead, it is better understood as an opportunistic organism within the genitourinary microbiome, meaning that under certain conditions, it may contribute to symptoms, but its presence alone does not automatically mean disease. And this highlights a very broader point about microbiology and women's health. This is the part that's super important because the vaginal ecosystem is not a simple battlefield of good bacteria versus bad bacteria. It is complex, it is dynamic, and it is a community of very different types of microorganisms that are interacting with hormones, the immune system, and environmental factors. So organisms like Ureaplasma remind us that context matters more than anything. The same microbe that can exist peacefully in one environment can become problematic in another. And understanding that nuance is important for both patients and clinicians because it helps prevent two extremes, either A, someone like a physician ignoring Ureaplasma entirely, or B, assuming that every single positive test means that something is wrong. The truth, as it is often in biology, is that this sits somewhere in the middle. And this is super important to understand so that you know how to ask the right questions and you know how to advocate for yourself. So if you do end up doing a comprehensive vaginal microbiome test like Juno Bio or Evi Bio or something of that nature, just a very comprehensive microbiome test, if you show up positive for Ureaplasma, it is not something that's necessary to treat if your vaginal microbiome is balanced. If you have a dominating presence of lactobacillus, that would be like me telling you, if you have a dominating presence of lactobacillus, but you have some Gardnerella in your vagina, go take antibiotics. That is so harmful. That is a terrible recommendation. And if anybody's recommending you to do that, that's really sad. They don't understand microbiology. Having the presence of some of these bad guys that society tells you about or the internet tells you about doesn't automatically mean that you need to get it treated because again, context matters. If your vaginal microbiome is generally balanced, it has a dominating presence of lactobacillus and has diversity, and you're not having any symptoms, there's no need to treat it. On the flip side, if you are someone who keeps getting positive tests for BV, you keep getting positive tests for AV, and these pathogens or these microbes, I don't like to call them pathogens because pathogens are something like chlamydia, gonorrhea. These are like foreign invaders. Those are pathogens. These guys are opportunistic. They're not necessarily bad. So that's why I like to be careful when I call something like a pathogen. But these opportunistic bacteria or these opportunistic microbes, they can exist quietly in the background and people get tested for the traditional things like we talked about, BV, STIs and things like that. And they keep getting BV, or even with a UTI, they keep getting UTI, and it's coming back for E. coli, but Ureaplasma is not being checked for. And so it goes kind of unnoticed. And so the antibiotics you're getting prescribed are treating a totally different type of bacteria like Gardnerella for BV or like E. coli for a UTI. And Ureaplasma is getting left untreated. And so your symptoms are persisting, and that is why you're still experiencing some issues. It definitely has to be an approach from both sides. If your physician is not regularly advancing themselves in this rhetoric, in this language, in this new information, and quite honestly, a lot of physicians just go off of what they learned in their education 10 years ago, 20 years ago, however old your physician is. So a lot of times they're not aware of the new research, the new data. There's no model really out there that makes physicians retake a test to re-update themselves or update themselves on new information. So many physicians, quite honestly, might just ignore Ureaplasma or just know from their education 20 years ago, 10 years ago, that Ureaplasma is a normal microbe that exists in the genitourinary tract. So they're going to say, why would I test for it? No, you're good. And I do see that online, that I see some microbiologists and I see some people on the internet saying, why would we ever test you for Ureaplasma? Ureaplasma is totally normal to have in the vagina. You don't need to be tested for it. So stop asking us to test for it because it's unnecessary. I disagree with that as someone who actively studies this, because I feel like you should at least look at the microbiome as a whole. And that's actually a gap in clinical settings, is that the vaginal microbiome is not assessed as a whole. You're either quickly swabbed for BV And I'll give you a quick little story time. So I actually am someone who regularly does comprehensive EVI testing. I swab my vaginal microbiome, whatever, and get it tested. And it always comes back as like a dominating presence of lactobacillus and things like that. But there's a presence of other microbes like Gardnerella, which is associated with BV, but in very small amounts, like let's say 3%, 2%. Very normal, small amounts of it, but a dominating presence of lactobacillus. It's funny because I could go to the doctor and Gardnerella will show up positive on a swab and has, and they'll recognize that as, oh, you have BV, because they're not assessing the whole microbiome. They're not saying, Gianna, like we need to look at lactobacillus. They're not looking at lactobacillus counts in the clinician's office. And that's why I think it's a little outdated with how vaginal microbiomes are swabbed and how we're looking for genitourinary infections. So I think that needs to be updated. That needs to be changed in a clinical setting, because if we're just looking for the presence, of course, like I always argue, just because you test positive for Gardnerella does not mean that you have BV. It's the same thing with ureaplasma. You can test positive for ureaplasma, does not mean that you have ureaplasma infection or that it's a disease or pathology. So that's really important to understand. And it can cause a lot of health anxiety when people are on the internet telling you it's an STI, or that, oh my God, if you have ureaplasma, you need to treat it. It is a normal commensal opportunistic microbe, but it doesn't necessarily mean that it's harmless, because given the right environment, it can cause harm. And that is why context matters. I hope that this video was incredibly helpful. Let me know if you guys found it helpful. And if you do want a part 2 or what else you want me to talk about for ureaplasma. But I hope you guys enjoyed this. I will see you guys in the next episode. Make sure that you hit subscribe and you're following us on all of our different platforms. I will see you guys next week. Bye, guys.